Hi students, in today's class we will be discussing on the sexual reproduction methods in bacteria. In bacteria, as you all know, there is no sexual reproduction. Actual sexual reproduction is absent here because and as a result there will be no genetic variability which is actually attained through sexual mechanisms. So it is difficult for them to produce a genotypic or phenotypic variations, but still these all variations occur in bacteria, and they obtain these type of variations through mutations, and genetic recombination and genetic variations are also attained by them. Then how is this possible if they are not having actual sexual reproduction as found in higher organisms? There are certain mechanisms, and these gene transfers are accomplished by parasexual mechanisms. Okay, so we have to study three mechanisms which are involved in bacteria. So actually bacteria have this parasexual, three types of parasexual mechanisms are there in bacteria. They are transformation, transduction and conjugation. So as of now we will be going through bacterial transformation only. So before that we have to know something. Some facts are there. That is one, I have already mentioned that there is no actual sexual mechanisms or actual sexual reproduction as found in higher organisms in case of bacteria. So also actual karyogamy and plasmogamy as found in higher organisms are also absent in bacteria. So they never produce zygotes. Zygotes are not produced in bacteria, but instead they only produce partial diploids. Okay, so only partial diploids are produced and they are called as merozygotes. So remember the term merozygotes. You may be getting this question for your 1 mark questions. So please note that merozygotes. In bacteria, they don't have the production of zygotes. So zygotes are not produced, but they have partial diploids which are called as merozygotes. And the original genome of the recipient cell, the cell which is receiving a foreign DNA, its genetic genome is called as endogenote. So the recipient cell's genome is called endogenote, and the foreign DNA or the fragment which is being received is known as exogenote. So we have learnt three new terms. First one was merozygote, second one was endogenote, third one exogenote. And when this exogenote gets integrated into the recipient cell's genome, then that recipient cell's genome becomes recombinant, and we call it as recombinant genome. Okay, so moving on to the first parasexual method, that is transformation. In transformation, it is the process wherein a foreign DNA fragment from a donor bacterium enter into a recipient bacterium and it gets integrated with the recipient DNA. And here what happens is a donor cell, this is a donor cell, the cell which acts as donor cell, it breaks down, and as a result when it breaks down it releases its genome. The DNA of the recipient cell is broken down into fragments, and that fragments gets released to the medium where these bacteria are growing. So in this case the donor cell breaks down and releases its DNA fragments to the medium. These DNA fragments are called as transforming molecules. These small fragments, as you can see, these are the they are called transforming molecules, and these are the exogenote as we have mentioned earlier, and this one is the endogenote because this is the recipient cell and the genome of recipient cell is known as endogenome. The genome which is being received is called as exogenome. So these fragments are released into the medium wherein the bacteria are growing, and then what happens is the DNA fragments gets absorbed. As you can see here, this DNA fragment gets absorbed, not absorbed, it gets adsorbed to the cell wall or the surface of the recipient bacterium, particularly at certain receptor sites or certain entry sites are there wherein certain receptor molecules will be present, and these DNA will interact with the receptor molecules at the entry sites, and they get into the bacterium and become integrated with the recipient genome, forming a recombinant DNA. So here also you can see, here donor cell is not shown in this picture. A fragment comes and approaches a recipient cell. It interacts with the receptors particles or receptor molecules at the cell surface, and from there through the entry point they gets into the cell, and after getting entry into the cell they get integrated with the genome of the recipient cell. So it becomes a recombinant cell. And in some cases it is not necessary that this foreign fragment or the foreign DNA get integrate into the genome always. In some cases they remain as independent molecules and they form plasmids. Plasmids are extra chromosomal independent DNA. So they may also remain as plasmids in the recipient cells. So the fate may be true. Either they become integrated or they may be existing as plasmids. So this is all about transformation. This actually produces a stable and heritable variation in the recipient cell. And it was at first described, this process was first described by Griffith in 1928. And now in these days, this process is actually being employed in certain genetic engineering procedures in lab conditions to artificially produce these transformed organisms. And it is not necessary that all bacterial cells can receive a donor DNA and undergo this type of transformation. All cells cannot accept foreign DNAs, and those cells which can accept are called competent cells. And this competent, we call that procedure as, or that property as, competence. So it is not necessary that every bacterium is ready to accept foreign DNA. Those bacterium which are receiving that foreign DNA particles, they are said to be competent. So this is all about transformation.